Hello everyone, this is Medical Gateway, and in this part we are going to study regarding the mechanism of action of antibodies. So antibodies can act via two mechanisms. One is the direct action. The antibodies directly attack the foreign invaders, and the second one is the action of complement system. So antibodies act via two ways. Number one is by directly attacking the foreign invaders, and second one is by activating the complement system, and then complement system basically attacks the foreign invaders. So in the direct action, there are four mechanisms by which antibodies attack the foreign invaders. So first one is the agglutination. So in the agglutination, the antigens are bound together by antibodies in a clump. So antibodies attach to the antigen, and then they form a clump of antigen and antibodies just like this. So these red ones are the antibodies, and then this one, the big ones, are the antigens. Now, as you know, there are two binding sites on this antibody. So one antigen can be attached to this binding site, and the other antigen can be attached to this binding site. And this antigen can then can be attached to more than one antibodies. So this antigen is then attached to this antibody, and this antibody. So this is how the antigens and antibody will form a clump. Okay, and then when this clump is formed, then this clump can be precipitated out. So second mechanism of direct action is precipitation of the antigen antibody complex. So this is a clump, or this is the antigen antibody complex. Now this antigen antibody complex becomes insoluble, and this complex just precipitates out. Okay, so when there is a clump, then it means that this antigen cannot go and attack the normal body cells. And when this whole complex is precipitated out, then it also means that this antigens cannot now attack the normal body cells. Now the third mechanism is neutralization. When the antibodies attach the antigen, they basically cover the toxic sites of the antigenic agent. Now this is an antigen. So for instance, if this is the toxic site of the antigen, this toxic site is covered by the antibody. So when the toxic site of antigen is directly covered by the antibody, then this antigen cannot be toxic to the normal body cells. So third one is the neutralization. What exactly is neutralization? That antibodies neutralizes the toxins or the toxic effect of the antigen. How they neutralize them? By covering the toxic site of antigenic agent. Okay. And the fourth mechanism is the lysis. Okay. So antibodies directly rupture the foreign bodies. They directly go and cause the lysis of the foreign bodies. So the four mechanisms are: one is agglutination, where the clump is formed, then the precipitation, that complex becomes insoluble, it is precipitated out, then there is neutralization, in which the antibodies cover the toxic sites of the antigenic agent, and then we have lysis, where the lysis or the rupturing of the foreign bodies take place with the help of the antibodies. Okay, so this all is the direct action of antibodies in combating the body against the foreign invaders. Okay, now then we have the second mechanism is the activation of complement system. Now, what exactly is a complement system? So complement system is basically 20 proteins, and these proteins are not enzymes, these are basically enzyme precursors, which means that these 20 proteins, when activated by antibodies, these 20 proteins or enzyme precursors will be converted into active enzymes, and then these active enzymes will attack the foreign invader by these all mechanisms. Okay, so let's first discuss that how antibodies can activate these enzyme precursors, and these enzyme precursors are basically forming the complement system. So there are 20 proteins which are enzyme precursors which form the complement system. Antibodies will come and activate these 20 enzymes, these 20 enzyme precursors, and they're activated by a classical pathway. Now, what exactly is classical pathway and how all the enzyme precursors are converted into active enzymes and how they are going to attack the foreign invaders, we're going to discuss in this slide. So this is the whole classical pathway of complement system. Now, what exactly happens? The story starts from here. Whenever the antigen enters the body, the antibody forms a complex with the antigen. So this antigen-antibody complex will stimulate the first conversion of one enzyme precursor into its active form. So we have C1 enzyme precursor. The C1 enzyme precursor will be converted into activated C1 enzyme. And who will convert it? Antigen-antibody complex will convert it. And now this activated C1 will activate C4 and C2 enzyme precursors, and they will be converted into their active form C42 plus and C4a. And then it is just like a chain of reaction that one activated enzyme will activate its next in the line enzyme precursor, and that the activated form of that enzyme will activate the other. So C1 is converted into C1's activated form, and that will convert C4 and C2 into their activated forms. And then C42 will convert C3 into its activated form, C3a and C3b. And C3 will convert C5 into its activated form, and C5 will convert C6 and C7 into their activated forms. And finally, we have C5b67 converting C8 and C9 into their activated forms. Okay, so these all are written on the left hand side of the reactions are the enzyme precursors C1, 2, 3, 4, 5, 6, 7, 8, 9. So these are inactivated enzyme precursors, and these are their activated forms C1 dash, C42 dash, C3b, C3a, C5b, C5a, and everything. Okay, so finally we have after the activation of all, the chain ends at C5, 6, 7, 8, 9. And between 5 and 6, there is a B. C5b, 6, 7, 8, 9. So this complex is known as a lytic complex. Why this is known as the lytic complex? Because this will cause basically the lysis of the cells, lysis of the foreign bodies. Okay. Now these activated enzymes will carry out the functions discussed here, by which the foreign invaders will be attacked by the complement system proteins. Okay, now, so first of all, these complement proteins activated by the classical pathway will cause lysis of the cell. So lysis of the cell will be done by lytic complex, as obvious from its name. So the lytic complex is basically C5b6789, the activated form C5b6789. Okay. Now you have to focus on these two ones, which I have highlighted, one in pink and the other one in green. So what is the function of this C5a? So function of the C5a is that it is involved in the chemotaxis of white blood cells. Okay. When we discussed in inflammation the chemotaxis of white blood cells, at that moment I told you that there are some complement enzymes which will play the role of chemotaxics, that will attract the white blood cells towards the site of infection. So among all the complement proteins, C5a is the one which will cause chemotaxis of white blood cells. Then we have C3b. Okay, now we also have discussed a concept which was opsonization of bacteria. So what exactly is opsonization of bacteria? So opsonization is basically that in opsonization, the foreign invader is tagged by C3b. C3b tags the foreign invaders. Now, what is the significance of this tagging? Whenever any foreign body is tagged by C3b, then the phagocytes will easily identify this foreign invader as the one to be engulfed and eaten up by. Okay, so it's basically there are foreign bodies, so the phagocytes now basically go and try to hunt these foreign bodies. So the tagging of the foreign body with C3b makes it easier for the phagocytes to recognize them and attack them and eat them up. So this process is known as opsonization. So opsonization is basically done by C3b. So up till now we have discussed three things. One is latex complex, which will cause lysis. Then we have C5a, which will cause chemotaxis of white blood cells. Then we have C3b, which will cause opsonization of bacteria. Okay, and then at the last we have C3, C4, C5, C3a, C4a, C5a. These all collectively will cause activation of mast cells and basophils. Okay, and you know what will happen by the degranulation of the mast cells and the basophils? They release a lot of pro-inflammatory substances causing the inflammation. Okay, so there are four things. One is latex complex, C5b, 6, 7, 8, 9. Then we have C5a causing the chemotaxis of white blood cells, C3b causing the opsonization of bacteria, and then C3a, 4a, 5a, they're all causing the activation of mast cells and basophils. Okay, now one interesting point is that microorganisms are also activating this C3 into C3b and C3a. Then what is the significance of this? Basically, C3b is causing the opsonization of bacteria, causing the tagging of bacteria for the phagocytes so that they can be easily recognized by the phagocytes. So the microorganisms themselves are activating this process of formation of activated C3b, and this activated C3b will tag these microorganisms, and which will result into their phagocytosis. Okay, now let's discuss all these points in this table. So interestingly, first three things which are done, first three mechanisms of actions which are completed by this activation of complement system are agglutination, neutralization, and lysis. So agglutination is the same agglutination which the antibodies were performing directly. Now, antibodies, how the antibodies were doing agglutination? Antibodies were forming a clump. Antigen antibody complexes were forming a clump. But how this complement system is going to do agglutination? Now there are complement products which will make the organism sticky, and then this foreign invading organism will stick to each other. So this complement products basically make invading organism stick to each other, and when they will stick to each other, they will agglutinate, and this is known as agglutination. Okay, then neutralization. So we also studied neutralization here, where the antibodies cover the toxic sites of antigenic agents. Now here in this case, the neutralization is done by complement enzymes. So basically complement enzymes attack the toxic structures of invaders. So there is a foreign body and it has a toxic part which is going to cause toxicity. So the complement enzymes will go and directly attack those toxic structures of the invaders. Then finally, the lysis were done by the antibodies directly. The lysis is also done by the complement proteins. And which one? We have just discussed. The lysis is done by the latex complex, the C5b, 6, 7, 8, 9. So this latex complex will go and cause rupture of membrane of the foreign invader. Okay. And then we have opsonization, as discussed. that opsonization is caused by C3b. C3b basically tags. It will tag the foreign invader so that phagocytes easily identify them. So C3b tags foreign invaders to get them phagocytosed by phagocytes. Then we have chemotaxis. Chemotaxis is very necessary for the neutrophils and macrophages, because by chemotaxis, these are attracted to the site of the infection. So chemotaxis is done by C5a, which will attract the neutrophils and macrophages to the site of infection. Then in the inflammation, mast cells and basophils are very important. So mast cells and basophils are activated by C3, 4, and 5a. And the mast cells will then release histamine and heparin, which will result in inflammation. Okay, and finally, by all these, the complement systems are causing the inflammation. And as you know, the inflammation will result into increased blood flow, increased permeability, which will bring a lot of WBCs, white blood cells, to the point of infection. And inflammation will also wall off this whole process of infection and the attack of the WBCs on the foreign invader. Okay, so this is how actually the antibodies will attack the foreign invader. So we are studying basically the immunity, and in immunity we have cell-mediated immunity and humoral immunity. So one is related with the B lymphocytes, others are related to the T lymphocytes. The B lymphocytes basically produce the antibodies. Okay, so antibodies, so these antibodies attack the foreign invaders by these mechanisms, the direct mechanisms, and by the activation of the complement system. Okay, so this was the mechanism of action of antibodies. See you in next part.